together?
It's just this particular example that they started off joined together and at the end they were separated. But different reactions are gonna do different things, right? Plants were doing photosynthesis, they'd start with individual subunits and then when we were done, they'd be joined together, right? And then we eat the sugar and we digest it into individual pieces. So it just depends on what reaction is happening. All right, we said that enzymes make reactions happen faster, right? Your donut would be digested in a couple of weeks instead of a couple of hours if you didn't have enzymes to speed it up. How that happens, enzymes reduce activation energy. Every reaction has an amount of energy that has to be put in to get it to go. Okay, so activation energy is the energy you need to start the reaction. And without enzymes, it's really high. You need a lot of energy to make a reaction happen. If you've got an enzyme involved, you need less. That's why the reaction can happen faster because you don't have to put as much energy in to get it started. I think we'll do over here on this side Y. Let's say that we have a cell, a big bacterial cell. And chemical A is floating around over here. Chemical B is over here. And we want to do that reaction A plus B yields C plus D. Well, with no enzyme in this cell, chemical A and B are just floating around in the cytoplasm, just bouncing around in that water. And in order for the reaction to happen, we need A and B to find each other. So let's say they're just randomly bouncing around, and eventually, a couple of hours later, A and B do find each other, but B is sideways and A is up and down. Reaction doesn't happen. You gotta have A and B pointed in the right direction. So they fly apart, bounce around in the cell, a couple of hours later they find each other again. Now A is upside down, B is the right way. Reaction doesn't happen. They bounce away, a couple more hours go by. Now they're pointed in the right direction, but they don't quite get close enough together because a lot of the water in the cytoplasm is flowing around, comes in between them, bounces them apart. Okay. You can see how long it might take for A and B to find each other, pointed in the right direction, and close enough together for the reaction to occur. To make all of that happen, the cell would have to spend a lot of energy to get those two things next to each other, pointed in the right way, close together. Putting an enzyme in here, inside the cell, the active site has an affinity, an attraction for A and B. It's not quite a bond, it's not like a magnet, they're not sucking them in. But if chemical A kind of wanders by the enzyme, there's an affinity and attraction to the active site. It's going to get pulled in. When chemical B eventually wanders by, it's going to get pulled in. So it's much more likely that now you're going to have A and B sitting right next to each other, pointed in the right direction, close enough together that water's not going to get in the way. The reaction's much more likely to happen now than over there on that side of the board. So the amount of energy you need is less. That makes sense? Sometimes enzymes need a little help doing their job. So we've got cofactors and coenzymes. This particular picture of the cofactor is helping to form the active site. Right? This is our substrate, and if we took this yellow cofactor away, the active site wouldn't have the right shape for this guy to fit. So the cofactor in this example is helping form the right shape to the active site. The only difference between a cofactor and a coenzyme is whether or not it's organic. Okay? Coenzymes are organic, cofactors are inorganic. Okay? And so organic, just reminds you, is carbon bound to other carbon, right? Or carbon bound to hydrogen. Here's one of those subtle differences between humans and bacteria. There are some elements that bacteria can make for themselves and some that we can't. Bacteria can make all of their own cofactors, all of their coenzymes. As long as they've got food to eat, they can build everything they need. There are some things that we cannot make for ourselves that we absolutely have to get from our diet. Okay, so some of these cofactors and coenzymes we have to eat, they can always make their own. Advantage bacteria. Good with that one?
So the cofactor and coenzyme makes the shape for the active
In this
site?
particular example,
But they
but
have
that's
many
not
other-
always what they do.
Okay.
Yeah, sometimes they're electron carriers, I mean, there's a variety of functions.
Which one is organic?
Organic is coenzymes, cofactors are inorganic. questions?
And when they, A and B or C and D, they form, when they form a bond,
Mhm.
what bond is that?
Depends on the reaction, depends on what C and D are. It could be ionic, could be polar or non-polar, just depends on which reaction we're doing.
Okay.
Now we're being intentionally vague with the A, B, C, and D there, to cover all the bases. Any other questions? Controlling enzyme activity. Okay, so we've got these enzymes inside of bacteria, but we don't need them all the time. Let's say that this is a bacteria in your intestines, and this enzyme is for breaking down lactose, the sugar in milk. Now, if you didn't have any milk today, do the bacteria in your intestines need their enzyme for breaking down milk right now? No. So they may temporarily shut that enzyme off. Like turning off the light in the room you're not in, right? You don't need it right now, save that energy. Okay? But then if you drink a glass of milk when you get home, they're gonna want this enzyme to be functional, so they'll turn it back on. That's regulating enzyme activity, turning them off when you don't need them, turning them back on when you do. Okay, so it's a reversible process. All we're gonna go through is a couple of ways to do that. Turn enzymes on and off as necessary. Allosteric regulation, this is our first method. Allosteric, that word by itself, means changes shape. We're gonna change the shape of the enzyme to turn it on and turn it off. So you may have noticed when I'm drawing my little enzyme, I made another little spot, right? If this were a ball, this would be kind of like the Pillsbury Doughboy, you stuck your finger in, right, and made a dent. That position is the allosteric site, used for allosteric regulation. Little indentation. Right now, there's nothing there, it's empty. So the active site has the proper shape, the substrates combine, the reaction can happen. So this, we're gonna consider for this enzyme, it's turned on right now, right? The active site has the proper shape, the reaction can occur. If we were to put something in that allosteric site, attach what is called an effector molecule, right there. The effector molecule attached. What that will do is change the shape of the active site. Okay, so when the effector molecule is attached, the active site has changed shape. Will A and B be able to fit into that active site now?
No.
No. So will the reaction happen?
No.
No. The enzyme is now effectively off. Attach the effector molecule to the allosteric site, that changes the shape of the active site, so the reaction in this case does not occur.
The same with that, the bonding could be different,
Right.
different types of bonding.
Mhm. Depending on which enzyme, which reaction we're talking about. Okay, so again, the online class doesn't get to see me draw, so this is their picture. Nothing in the allosteric site. The active site is the right shape for this substrate. You put the effector molecule in, which unfortunately is sometimes called the allosteric inhibitor, same thing as effector molecule. You attach that, the active site has changed shape, so now our substrate can't bind. Reaction won't happen. Completely reversible. You take that effector molecule or take the inhibitor out, the enzyme's gonna go right back to looking like this. The shape will change again. Second way to regulate enzyme activity, turn them on and turn them off. Feedback inhibition. Feedback inhibition is 99% the same as allosteric regulation. Everything we just said is still true. We're gonna have an effector molecule attaching to the allosteric site, changing the shape of the active site. Everything we just said for allosteric regulation, also true for feedback inhibition. The only difference with feedback inhibition is what this effector molecule is. In feedback inhibition, the effector molecule is one of the products of the reaction. The effector molecule that binds and changes the shape of the active site is one of the products of the reaction. So we come back over here to our cell. We put an enzyme in here. Right, we've got chemical A and chemical B floating around. And probably multiple copies of them. lots of A and B floating around. We haven't done the reaction yet. There's no C or D inside the cell. The enzyme has nothing attached to the allosteric site, so it's the right shape. A and B are gonna bind. The reaction's gonna happen. C and D are gonna be produced, right? Over and over and over. So the levels of C and D are gonna start to build up in the cell. Once there's a lot of chemical C is gonna come back over and bind to the allosteric site of the enzyme. And that's gonna change the shape of the active site. So is the reaction gonna happen now? No, it's a self-regulating reaction with feedback inhibition. One of the products of the reaction acts as the effector molecule shut it off. Whereas plain old allosteric regulation over here, this effector molecule was some other random molecule in the cell. It was not a product of the reaction.
And we have to remember how to draw that, right?
Well, I'm not gonna ask you to draw it. You have to be able to explain it. If it helps you to draw it, you certainly can, but no, you'll never get a question that says, draw an enzyme and show me allosteric regulation. No, if you were to get a short,
And then the yield time.
not necessarily. I mean, the kind of question where you'd have to write anything out about this would be something like, describe allosteric regulation in enzymes.
Oh,
And then you could just say, the effector molecule attaches to the allosteric site, that changes the shape of the active site, and the enzyme doesn't work. Okay, so you won't necessarily have to draw it. If it helps you to explain it, you can. The difference between feedback inhibition and allosteric regulation, make sense? Good with that? All right. Those are both, wait, before I leave that, those are both types of non-competitive inhibition. Okay, both allosteric regulation and feedback inhibition are non-competitive, which I think I have in your notes. Yeah, you go back to this slide, down here, non-competitive inhibition. What that means is that our effector molecule here does not compete with the substrates for the active site. They're not binding to the same place. Okay? The effector molecule attaches to the allosteric site, the substrates to the active site. So non-competitive inhibition. The effector molecule and substrates are not competing for the same location. Which means that there is a different type that is competitive. So this is our third way to regulate enzyme activity, to turn it on and off. Competitive inhibition. Now we are going to have a molecule that is competing with the substrates for the active site. And this molecule is usually called an inhibitor. The inhibitor is going to bind to the active site, the big I in there for inhibitor, and block it. So in this one, we're not changing the shape of the enzyme at all. There's nothing attached to the allosteric site. The active site has the right shape, but the inhibitor binds to the active site and blocks it. So the substrates can't bind. So it's competitive. The inhibitor is competing for the active site with the substrates. This one is typically not reversible. Once that inhibitor is in there, it's usually in there forever. Why?
I mean, like, why would that happen?
That usually doesn't happen on purpose. I mean, it's just not something typically that humans or bacteria do of their own volition. And there are probably a few examples where they do, but most of the time it's a poison, something that's going to kill you or kill the bacteria, gets in there and blocks up the active site so the enzyme doesn't work.
So, it's something like cyanide.
Well, along those lines.
I mean, just like.
Yeah, just to give you an idea. Yes, ma'am.
So is that why when there's certain medications that they're advertising on TV, if you're taking such and such inhibitor, don't?
Oh, no. There's so many possibilities for what that could be. Usually the inhibitors are another class of drugs that are doing something else. Yeah, so it really probably doesn't have anything to do with enzymes.
Okay.
Yeah, it's just affecting the function
Cause you
of
got a poison.
the other drug they're on.
I'm
So
weird.
questions on competitive inhibition.
I'm the same thing.
Whew. All right, so we're done with the ingredient list now. This is just sort of summing it up. These are all of the things that you have to make metabolism happen. ATP, energy source and electron source,